Welcome to the deep dive. We're going to map out the most significant pathogens affecting cannabis sativa L. We'll help you understand exactly how environmental conditions can make a perfectly healthy plant suddenly vulnerable, and teach you how to recognize those crucial symptoms at every single growth stage. We'll also explore the fascinating idea of disease-resistant varieties and crucially arm you with cutting-edge integrated strategies to keep these valuable plants thriving.
When we talk about growing cannabis, especially in a controlled greenhouse environment, it's way more than just getting the light nutrients right. There's this whole unseen world operating, like a silent battlefield of microbes and diseases, always there. And these can dramatically impact both the quality and the yield of your crop, massively. So to start us off, who are the main players in this hidden war? Which pathogens should growers really be concerned about?
Yeah, that's a great starting point. And what's really critical to understand here is just how diverse these threats are. And importantly, they often don't operate alone. You can get several of these on the same plant causing trouble together. Among the most important diseases we consistently see, especially in greenhouse cannabis, are those nasty root rots. They're primarily caused by Fusarium and Pythium species. Then there's the infamous bud rot, which is mainly from Botrytis cinerea, that one's notorious, and of course the very widespread powdery mildew that comes from Glovomyces ambrosia. You see that one a lot.
Okay, root rot, bud rot, powdery mildew.
Yeah.
Those sound like the big three.
They are major ones, yeah. But the list doesn't quite end there. We also have to contend with something called Cannabis stunt disease. It's actually caused by a microscopic saboteur, the hop latent viroid, or HLVd for short.
A viroid? What exactly is that? Is it like a virus?
Sort of, yeah. Think of HLVd as this rogue, microscopic piece of genetic code. It doesn't even have a protein coat like a virus. It basically hijacks the plant's growth machinery. And the really tricky part, often with no obvious symptoms until it's way too late for the crop. Very sneaky.
Wow, okay. That sounds difficult to manage.
It can be. And then even after you harvest, you've got another layer of challenges, a whole range of microbes, including various yeasts and molds like Penicillium, Alternaria, Cladosporium, Fusarium, and Aspergillus. They can all significantly reduce the quality of the final product.
So even when you think you're done, you're not quite done.
Exactly. Now, here's a really crucial insight for cannabis cultivators, something that makes it different. Unlike a lot of other crops where you might reach for synthetic fungicides, those aren't typically allowed here. Only reduced risk biological and biorational products are permitted.
Reduced risk. What does that mean in practice?
It means they're primarily protective, not curative. They help prevent infection, but they don't usually wipe out an existing one like some synthetics might. This fundamental difference. It forces growers into a highly proactive, preventative mindset. You almost have to act like a plant bodyguard rather than, an emergency responder.
Ah, I see. That preventative mindset, that sounds like a real game changer. Less reacting, more anticipating. So, okay, if these are the unseen enemies and you have to be preventative, how do you even spot them early? You mentioned cannabis goes through different growth stages, stock plants, flowering, etc. Do the symptoms change? What specific clues should we be looking for at each step? It sounds like we need to become like plant detectives.
You absolutely do. Becoming a skilled plant detective is essential. And yes, understanding how symptoms show up at different stages is totally key for early detection and, well, effective management. Let's break down those clues stage by stage. Okay, first, in stock cultivation. We're talking about your mother plants here, the ones you take cuttings from. You might just see a general decline in growth. Maybe the plant seems less vigorous than it used to, generalized leaf yellowing, or even poor root development, especially in older plants. These could be signs of Fusarium or Pythium infections in the roots.
Okay, so subtle signs mostly.
Often, yes. Now, HLVd, that viroid we mentioned, it's particularly sneaky here. It often remains largely asymptomatic, meaning no obvious outward signs at all on the mother plant. But occasionally, you might catch some subtle curling or maybe some mottling, like uneven color, on the youngest leaves. If you were to actually cut into the stem, seeing internal discoloration in the pith, that's the spongy center, and the xylem tissue, the water transport part, that's a classic symptom of Fusarium.
Right, you'd have to look inside the stem itself.
Yep. And just general root browning, that could point to either Fusarium or Pythium. Powdery mildew is usually more obvious. It shows up as these clear, white, often kind of circular fungal colonies right on the upper surfaces of the leaves. Hard to miss once you know what you're looking for.
Okay, that makes sense for the mother plants. What about when you take cuttings? The propagation stage.
Right, propagation. So If your stock plants are infected, even asymptomatically with HLVd, then poor root growth on the cuttings themselves is a major red flag for that viroid. The cuttings just won't root well. You'll also see damping off symptoms, especially from Fusarium species, and particularly in genotypes or strains that are susceptible.
Damping off, what does that look like?
It's pretty grim.
Yeah.
The seedling or cutting stem basically just collapses and rots right at the soil line. It just flops over. And remember, the high humidity you need for rooting cuttings. While it's good for rooting, it unfortunately also creates the perfect breeding ground for gray mold, Botrytis cinerea, and also common saprophytic fungi like penicillium.
Saprophytic.
Yeah, those are fungi that feed on dead or decaying organic matter. So if there's any dead tissue, they can jump in.
Got it. High humidity is a double-edged sword there. What about vegetative growth after rooting?
Good question. During the vegetative growth stage, those root-infecting pathogens like Fusarium and Pythium, they just keep developing if they're present. Symptoms here can include that persisting yellowing of leaves, stunted overall plant growth. The plant just isn't sizing up like it should. You might see browning of the roots if you check them, and in really severe cases, you can even lose the whole plant. Plant death. HLVd, the viroid, can also really hold back the plant size here. The plants might look otherwise okay, just much smaller than their healthy neighbors.
Okay, so stunting is a key sign across multiple issues then.
It definitely can be, yeah.
Now the flowering stage, that feels like where the stakes get really high, right? For the final product.
Yeah, absolutely. The flowering stage is where things get truly critical, economically speaking, because that's when the valuable buds are forming. Any root infection symptoms from earlier stages, like that leaf yellowing, wilting, stunted growth, they often become really pronounced now, much more obvious. HLVd symptoms, if they somehow went undetected before, typically show up about 1 to 3 weeks into the flowering cycle, and they present as significantly reduced inflorescence size. Growers might call it dudding or stunting or dwarfing of the buds. They just don't develop properly. You might also see yellowing of the bract leaves, those little leaves within the bud structure, and just general stunted plant growth overall.
That sounds devastating for yield.
It absolutely is, and powdery mildew development. That can also become far more severe during flowering, especially in susceptible genotypes. It can end up covering the leaves and even growing onto the developing buds themselves.
Ugh, mildew on the buds. Not good.
Definitely not. And then, as you get closer to harvest time, Botrytis cinerea bud rot becomes highly likely to appear, especially if conditions are right. That stuff can just devastate quality and yield because it literally rots the buds, often from the inside out. You might not even see it until it's too late.
Wow. Okay. And even after harvest, you said there are issues.
That's right. Post-harvest concerns. These aren't really diseases on the living plant anymore, but they're all about the quality and safety of the dried product. The colonization of those harvested inflorescences, the buds, by various yeasts and molds we mentioned, Penicillium, Alternaria, Cladosporium, Fusarium, Aspergillus, it's actually very common. These microbes can seriously affect the final product's quality, its aroma, its safety. And often, these issues are only detected through rigorous lab testing that's required before the product can be sold.
That's an incredible breakdown of the symptoms across the whole life cycle. It really highlights how vigilant you need to be. It sounds like even with really meticulous care, these problems often start subtly. They only become really obvious when they're quite advanced. So if early detection is such a challenge, does the environment itself kind of act as an accomplice? Does it make plants even more vulnerable before they show clear signs? How do conditions in a greenhouse actually make these problems worse?
You've absolutely nailed a crucial point there. Environmental conditions are a massive factor. Huge. They often act as the primary driver for disease outbreaks. For instance, something seemingly simple, like excessive waterlogging of the growing medium, keeping the roots too wet. That can lead directly to root browning and essentially suffocate the roots. That stress makes the plant significantly more susceptible to invasion by Fusarium and Pythium.
So overwatering is a direct invitation.
Pretty much, yeah. And we also see clear seasonal patterns playing out. Warmer temperatures combined with higher humidity, typical during summer months, can dramatically increase the incidence of these root-infecting pathogens. They just thrive in those conditions.
Yeah.
Conversely, cooler and more humid conditions, which you might get more often during winter, can really enhance powdery mildew development. It likes it cool and damp.
Interesting. So different seasons favor different diseases.
That's often the case. And think back to the propagation stage, that necessary high humidity we talked about for rooting cuttings. Well, it also creates that ideal breeding ground for the rapid spread of Fusarium and Botrytis cinerea.
Right, the double-edged sword again.
Exactly. Then, during the flowering stage, just the sheer increase in plant biomass, those dense, bushy canopies, that naturally leads to higher humidity levels right around the plants, within the canopy itself. And that tracked humidity can then actively promote both powdery mildew and that dreaded bud rot.
Makes sense. More plant, more moisture trap.
Yep. And even things like general root damage, maybe from transplanting or handling, or simply excessive watering and having localized flooding, like puddles on the floor or benches, that can quickly spread pathogen inoculum. That's the infectious material, like spores or bits of fungus throughout the whole growing area. It just intensifies the disease spread.
So water management is key, not just for root health, but for stopping the spread too.
Absolutely critical. And as you sort of hinted at, those overall seasonal environmental fluctuations, they directly impact the buildup of those post-harvest microbes we discussed, the ones found on the final product. The weather outside can influence the microbes inside.
Wow. So understanding these environmental triggers, it's like understanding the battlefield conditions, isn't it? Okay, given this, well, pretty daunting list of threats, and the fact that growers can't just spray heavy-duty synthetic fungicides, it really does sound like they're facing an uphill battle. Is there any kind of silver bullet solution? Or are we really talking about a much more integrated, like multi-layered approach to protecting these plants? What are the practical integrated strategies that actually work out there?
Yeah, you're right. There's no single silver bullet. And that's precisely because it's such a complex interaction between the plant, the specific pathogen, and the environment. They all play together. This is exactly where integrated disease management, or IDM, comes into play. It's not one thing. It's a coordinated, multi-pronged approach that combines a whole bunch of different tactics.
Okay, IDM, integrated disease management, makes sense.
Yeah.
What are the main pillars of that approach?
Well, first, and you could argue it's the absolute bedrock of prevention, is biosecurity and sanitation. This is your first line of defense, always. It means implementing really standard but strict practices. Things like using foot baths before entering grow areas, wearing protective clothing like coveralls or lab coats, and thoroughly cleaning all surfaces and equipment, and I mean everything. Gutters, tables, floors, drip emitters, pots, tools, everything. You'd use powerful disinfectants like products containing hydrogen peroxide with peracetic acid or maybe isopropyl alcohol.
So cleanliness is paramount.
Absolutely. It's also crucial to establish a strict quarantine protocol for any new plant material coming into the facility. Whether it's clones or seeds, you need to isolate these new plants maybe for 3 to 4 weeks and meticulously test them, often using those highly sensitive PCR or RT-PCR tests we mentioned earlier. They detect the pathogen's actual DNA or RNA. Only after they test clean should you introduce them into your main cultivation area.
Quarantine and test, got it.
And if you do find infected plants, either in quarantine or later on, promptly removing and destroying them is absolutely essential to break the disease cycle. You have to remove the source of the infection before it spreads further.
Eradicate the problem immediately.
Exactly. And speaking of eradication, it can be remarkably effective, especially for something like HLVd. There was a study showing that routine weekly testing of stock plants, followed by immediately removing any HLVd-infected ones, gradually reduced the HLVd frequency in that facility from a pretty high 22% all the way down to just 1% over about 6 months.
Wow, that's a huge reduction just from testing and removing.
It really is. So as a general rule, it's also good practice to replace your stock plants, your mothers, every 3 to 4 months with fresh, certified pathogen-free material, just to keep that starting pool really clean.
Okay, biosecurity, sanitation, quarantine, testing, removing infected plants. That's a strong foundation. What's next in IDM?
Next up is environmental management. This is about actively manipulating the greenhouse climate to make it less hospitable for the pathogens and more optimal for the plant.
Controlling the weather inside, basically.
Essentially, yes. So for the cloning or seedling stages, you'd want to maintain temperatures roughly between 20 to 24 degrees Celsius, that's about 68 to 75 Fahrenheit, with relative humidity kept high, above 90%, to help cuttings root. For the stalk and vegetative stages, you'd aim a bit warmer, maybe 25 to 28 Celsius, 77, 82 F, with humidity between 65% and 75%. This promotes vigorous growth. Then, during flowering, you typically shield, again, maybe 23 to 28 Celsius, 73, 82 F, but drop the relative humidity down to 50% to 70%.
Why the changes, especially the humidity drop in flowering?
Good question. These precise parameters help manage something called vapor pressure deficit, or VPD. VPD is basically a measure of the drying power or the thirst of the air. It influences how much water the plant transpires, breathes out, and also how it absorbs nutrients. Getting VPD right is critical for both optimal growth and for disease resistance. Lowering humidity in flowering helps reduce the risk of bud rot and powdery mildew in those dense canopies.
Oh, okay. VPD. That's a key concept then.
Very key. You can also do things like increase the interval between watering events. Let the growing medium dry out a bit more to minimize conditions favorable for root pathogens. Avoid those constantly soggy conditions. In the flowering stage specifically, increasing air circulation is huge. Aiming for about 0.5 to 1.0 meters per second of airflow using fans can significantly lower the humidity within the plant canopy and also physically help reduce microbial populations settling on the inflorescences.
More airflow, less moisture, fewer microbes.
Exactly. Reducing plant densities, meaning spacing plants out more, can also help improve airflow, though that might come with a slight trade-off in terms of yield per square meter.
A balancing act.
Always. And strategically, some growers might even alter their seasonal planting times. If they know, say, late fall brings conditions perfect for botrytis bud rot in their area, they might try to schedule their harvest before that period hits.
Clever. Working with the seasons instead of just fighting them. Okay, managing the environment sounds like building a fortress against these invaders. But what about the plants themselves? Can simply choosing certain cannabis varieties make a real difference? Does genetics play a role?
Yes, absolutely it does. Utilizing disease-tolerant genotypes is a very important piece of the IDM puzzle, and honestly, it's often underutilized. Now, we don't really have widespread, specifically bred resistance genes like you might see in, say, corn or wheat. Not yet, anyway, through traditional breeding in cannabis. However, there's huge natural genetic variation within cannabis, and that means some genotypes, some strains, just naturally show better tolerance to certain diseases than others.
So some are just tougher by nature.
Pretty much. For example, research has identified a genotype they called LB that showed really high tolerance across the board, good against powdery mildew, against hop latent viroid, against botrytis bud rot, and even against root pathogens.
Wow, an all-around tough one.
Yeah, relatively speaking. In stark contrast, other genotypes they tested, like ones called LO and MP, were found to be highly susceptible, especially to powdery mildew. So, consciously selecting and growing these more tolerant genotypes can make a significant difference, especially when you know you're going into a period of high disease pressure, like a particular season.
That makes a ton of sense.
Yeah.
Fight genetics with genetics.
Right. Then we move into using beneficial organisms, biological control agents, or BCAs. These are basically your reduced risk living allies. Often they're beneficial microbes like fungi from the Trichoderma genus or Gliocladium catenulatum.
Friendly fungi fighting bad fungi.
Exactly. Imagine them as tiny bodyguards for your plant's roots and leaves. They work in different ways. Sometimes they outcompete the pathogens for space and food. Sometimes they directly parasitize or attack the bad guys. And sometimes they even trigger the plant's own defense systems.
That's fascinating. How are they used?
They're excellent alternatives to synthetic fungicides, especially in preventative programs. They're particularly useful for preventing damping off caused by fusarium on cuttings. You might apply them as a drench or dip. They can also reduce mortality from fusarium and pythium in the vegetative stage when applied to the roots. And remarkably, research has shown that even in flowering plants, applying a product like Root Shield HC, which contains a specific strain of Trichoderma harzianum, not just to the roots, but as a spray onto the foliage and flowers, it actually helped reduce botrytis cinerea bud rot development. And it also suppressed other undesirable microbes like penicillium species in the harvested inflorescences. It even helped reduce powdery mildew, too.
Wow, spraying a beneficial fungus to fight a harmful fungus. That's clever.
It is. Other effective BCAs that have shown promise against bud rot include products like Regalia, which is actually a plant extract, double nickel, Lifeguard, and Prostope. There are several options now.
Good to know there are biological tools available. Are there any sort of safer chemical options too? You mentioned reduced risk earlier.
Yes, in addition to the biologicals, there are also some reduced risk chemical products available, mostly for foliar application, meaning sprayed on the leaves. For powdery mildew, for instance, preventative sprays like Sufoil X, which is essentially a highly refined mineral oil, can be effective. It works partly by suffocating the fungus. And Regalia max, that's an extract from the giant knotweed plant, Rhinutria saccharinensis. It doesn't kill the fungus directly, but actually triggers the plant's own natural defense responses, making it more resistant. It's called systemic acquired resistance.
Boosting the plant's own immunity. Neat.
Exactly. Now, for curative treatments, if you already have an outbreak of powdery mildew, products like Millstop, which is potassium bicarbonate basically, a form of baking soda, can be used, and Cyclone, which contains citric and lactic acid. These often work by physical means. They might alter the pH on the leaf surface, making it hostile to the fungus, or they can help desiccate or dry out the fungal mycelium, that network of fungal threads, to stop its growth.
So softer chemistries that work differently than traditional fungicides.
Generally, yes. They tend to have lower environmental impact and fewer safety concerns.
Okay, that covers managing the living plant. What about protecting the harvest? Those post-harvest microbes.
Right. Crucial last step. Post-harvest practices. This is all about handling the product correctly after cutting it down. Reducing the moisture content rapidly and thoroughly through proper hang drying in a controlled environment is key. Moisture is the enemy here. Also, many experts recommend trimming the inflorescences, the buds, only after they are fully dried. Trimming wet can potentially spread spores. And of course, rigorous cleaning and sanitation of all processing equipment, trimmers, containers, surfaces is just as important here as it was in the grow room.
Consistency and cleaning from start to finish.
You got it. Now, in some cases, especially in highly regulated markets, if microbial levels still exceed the strict regulatory limits despite best practices, irradiation might be used as a final step. This involves using gamma rays or electron beams to essentially sterilize the product. It's effective, but it's also quite costly and sometimes controversial.
Irradiation? Okay. A last resort, maybe. Wow. What a truly comprehensive exploration of this whole complex world of cannabis plant health.
Yeah.
It's crystal clear that managing these plants effectively isn't about one magic trick. It demands constant vigilance and a really integrated, multifaceted approach, doesn't it? It really seems to be about combining that tight environmental control, leveraging biological allies, being meticulous about sanitation, and making smart choices about genetics.
That sums it up perfectly.
Okay.
Exactly. And it's important to remember that the research in this area is moving fast. It's constantly revealing new details, new challenges, and new avenues for protection. So for you, the curious learner listening right now, maybe consider the bigger picture here. Think beyond even these current integrated management strategies we've discussed. Imagine the potential down the road. What if we could fully harness the plant's own internal microbes, its endophytes, the beneficial microbes living inside the plant tissues, to naturally boost its resistance to pathogens even further?
Like internal bodyguards.
Kind of, yeah. Or what about pinpointing disease instantly, long before we can see it? Maybe using AI-powered cameras trained to recognize super subtle changes in leaf color or texture that signal disease onset. Or even developing electronic noses, sensors that could detect the unique volatile organic compounds, the specific smells that stressed or sick plants release, giving us a warning before any visible symptoms appear.
That's fascinating. Early detection using tech.
Right. These emerging technologies, combined with a deeper and deeper understanding of the plant's own defense mechanisms, they really point towards a future where disease management in cannabis cultivation could become even more precise, much more preventative, and ideally, even more environmentally harmonious.
It's really a testament to the dynamic nature of agricultural science, isn't it? And that continuous pursuit of knowledge, all aimed at growing healthier plants and achieving higher quality, safer products. Well, that brings us to the end of another deep dive. We really hope we've given you that shortcut to being truly well-informed about cannabis plant health, and maybe highlighted that intricate dance between the plants, their pathogens, and the environment they live in. The journey of knowledge, as always, is ongoing. We invite you to keep mulling over these fascinating insights. Maybe think about how these principles apply to other plants you grow or encounter. Thank you so much for joining us today. Stay tuned for our next deep dive.